So thank you, Dr. Gibson, for the colorful and positive introduction. My name is David Barfknecht, as Dr. Gibson said, and today I want to present the research I've been working on for my doctoral dissertation relating to diversity, community structure shifts, and patch characteristics in natural xeric forest openings within Southern Illinois. So the outline I'm going to go through this presentation basically follows the chapters of the dissertation as it is. And so I'll start out with a literary review and a hypothesis, work through all of my data chapters, and then I'll wrap up everything with some general conclusions that pull all those chapters together into a more collective framework. So the first thing I want to talk about is diversity, which continues to be a very large concern for a lot of different ecologists as well as land managers. And it's important to preserve it because of its importance for ecosystem stability, resilience, as well as function. And when we look at the different aspects of diversity, we can look at taxonomic diversity, which most of us here are quite familiar with, the number of the relative abundance of taxa in a set study system. And then furthermore, you can look at things like phylogenetic diversity, which relates to the evolutionary relationships between taxa. Or you could also look at functional diversity, which deals with the functional roles that taxa perform within an ecosystem, typically derived from functional traits. And these aspects of diversity are quite important for understanding the rules of community assembly and the mechanisms that drive them. So if we talk about phylogenetic diversity, at a more diverse end of the spectrum, phylogenetic assemblages tend to be more overdispersed, meaning that taxa are typically further distantly related than was expected by chance, where at lower levels of phylogenetic diversity, you tend to have clades within a system that are more closely related than expected by chance. But if we take it on to functional diversity, it's very similar, where functional overdispersion is within assemblages. The functional traits between these individual species within a site tend to be more variable than expected by chance, whereas clustering at the lower end of functional diversity, you see a lot of clustering of functional traits within a specific study system. So relating that to how it depends on community assembly, local ecosystems are a product of the regional species pool. And so you have these different layers, first of all, related to dispersal limitation, environmental filtering, which is usually related to abiotic conditions. And then the final filters we see at the lower part of this left screen deals with the competitive, it says competitive exclusion here, but it's really biotic interaction. So these things are not necessarily always competitive, but they can also be mutualistic. So as a result, due to these processes, local assemblages tend to exhibit variation in their phylogenetic and their functional diversity, where the typical rule of phylogenetic inertia here shows that we more often assume that species that are closely related also tend to be more functionally similar. However, there are individual cases where you can have functional surplus or functional deficiency, and those tend to relate to specific community assembly drivers. And to further note that there are distinct differences in what phylogenetic and functional diversity tell us about individual ecosystems, you can see that higher levels of phylogenetic diversity seems to indicate that there's some form of competition within specific clades, whereas low phylogenetic diversity is intercladal competition or some kind of a filtering mechanism that's driving a lot of that assembly. And then with functional dispersion, we see at higher ends of functional diversity than what are expected by chance, we can see some kinds of niche partitioning or limiting similarity between functionally similar species. And then at the functional underdispersion or low functional diversity end, there tends to be more of a pressure from different filters within the system. And another thing important about this diversity in assemblage is that external threats related largely to anthropogenic activities have been shown to predict that a lot of these changes between these different relationships are to vary within the near future, if it hasn't already, which I'm sure it has. So moving on to a more spatial aspect of this is looking at metapopulations and metacommunities, where metapopulations are these interactive yet spatially distinct populations which exchange individuals or genetic material between them. And those dynamics are largely dependent on the connectivity of individual patches, as well as the patch characteristics itself. As you can see in this top diagram, they're specifically looking at patch size. So larger sites closer together tend to have better species dispersal between them. Looking at a larger scale, looking at several populations, we look at metacommunity dynamics, and there are several different frameworks that talk about community dynamics, but the important part here is that they interact, the species populations interact with one another. And plants in general are actually quite a useful system for modeling these different spatial and using spatial analyses, as they maintain a largely sessile habit, excluding events such as pollination and seed dispersal for the most part. So my system that I worked with to look at some of these different dynamics were natural xeric forest openings, specifically within the Southern Illinois region. And a little history related to this is that pre-1800s, the most dominant communities in Illinois were tallgrass prairies and barrens, followed by hardwood forests. However, as European settlement went on and they practiced agriculture, a lot of the areas that were more suitable for farming practices and lumber harvesting, they were largely fragmented. And now we know that about 99% of the grassland communities within Southern Illinois are no longer remnant. And so these natural xeric forest openings are kind of separate from that, because they didn't, most settlers did not see that they were very suitable for, you know, having cattle fields. They had very low substrates, very, what almost appeared to be malnourished trees and stuff. So that those areas were typically not utilized. And so what has been found in previous research by Alice Heikens in 1988 is that most of these communities are largely driven by their edaphic soil conditions and are typically quite fire dependent in order to ward off woody encroachment. And so happily, a lot of these areas have been set aside as nature preserves or ecological research areas. However, even if they are protected, they still face the threats related to anthropogenic influences such as climate change and exotic invasions. So with my research, I wanted to basically elucidate how different patch characteristics affect community structure in these naturally patchy ecosystems by, first of all, looking at metapopulation and metacommunity structure within a local set of sandstone outcrop communities. Furthermore, looking at characterizing the phylogenetic, functional, and taxonomic phylogenetic and functional compositional shifts in a broader group of natural xeric forest openings that are not just sandstone outcrops. And then finally, I wanted to understand how diversity and environmental conditions in these communities are spatially structured and how they affect the biological turnover over when I considered surveys in 1988 and when I conducted my surveys in 2019. So my first chapter dealing with metapopulation and metacommunity structure, there were 4 hypotheses that were associated with this specific study. I wanted to see if species within these patches had metapopulation dynamics. I wanted to observe if there was any kind of a significant non-random metacommunity structure associated with them. I wanted to see if there are correlations between diversity, functional traits, and patch characteristics across all the sites. And then finally, I wanted to see if basically taxonomic diversity indices, including total and metapopulation species per site, were driving the phylogenetic and functional diversity of specific or individual patches. So for this project, this took place at Jackson Hollow Ecological Area in Polk County, Illinois, and the surveys were conducted in 23 observable patches at the western extent of the ecological area. And within each of these 23 patches for 3 months during the summer, I collected species occurrence data including mosses and vascular taxa within each of these sites. And then thanks to Nick Seaton and Kevin Rowling for coming out and helping me actually get some drone imagery so that with that, I could specifically isolate those patches in a spatial fashion. And then all the communities were included in the Metacommunity and the phylogenetic analyses, but for metapopulation analyses, if any species were ubiquitous throughout the sites or they were singletons for a specific site, they wouldn't work for the model. However, out of all these sites, 130 taxa were observed across all those sites. And what's interesting to note right here is that the most specious, the most taxonomically diverse sites were actually the ones that were closest to Trigg Tower Road, as you can see to the left here. Just a general observation there. Moving on to look at metapopulation and metacommunity analyses, I used Hansky's incidence functional model, which is a spatially explicit model for species occurrences across these patches, and that assesses which species are significantly showing metapopulation dynamics. For the metacommunity analysis, however, I use this framework known as the elements of metacommunity structure, which is based on 3 separate indices. First of all is coherence, which basically analyzes how many species are consolable to a single site dimension. And if that is either negative or non-significant, we get these odd metacommunity structures here, or no metacommunity structure more rather. And then the next step in that is to look at turnover. This is biological turnover. And if that's negative or insignificant, you can get these kind of structures. These nested subset sites are reminiscent of certain kinds of island systems where one patch is supplying most of the immigration of other species to different patches. And then quasi-structure, there are several of these, but I collapsed it in this diagram for ease. And then finally, looking at boundary clumping, if there is a positive boundary clumping, that means that there's a Clementian metacommunity structure, whereas non-significant and negative boundary clumping indicates a more individualistic species gradient across patches. So there are several different natural empirical examples of these. And I've seen all of these different kind of metacommunity structures pop up from Brazil rainforests to German ravine forest ecosystems. So these are kind of good for kind of modeling the overall metacommunity structure. So the results from that is that out of the 130 species that were observed, 20 of those species had significant metapopulation dynamics. And out of these, 4 of them were composites, 5 of them were graminoids, either Cyperaceae and Poaceae. However, out of the 5 exotic species that were observed, 4 of them did exhibit this significant metapopulation dynamic. The only one that didn't make it was Lonicera japonica, which was ubiquitous across sites. And so looking at a chi-square analysis, it shows that exotic species that tend to be annuals or biennials are more likely to have metapopulation dynamics than expected by chance. However, the growth forms are not anything more than by chance, statistically speaking. And then when looking at the elements of metacommunity structure framework, all 3 of the elements were significantly positive, indicating that these metacommunities are exhibiting what we would call a Clementian structure. But Clementian is kind of an old way to describe systems like this. So really what these elements are telling us is that there is no checkerboard or random pattern between them. The species are regularly replaced across these sites, and species are tending to be replaced as groups as opposed to individualistic responses, which means that the species within these sites are actually reacting to an environmental gradient in a somewhat of a similar way across patches. So the next part of the next part that I did to look at phylogenetic and functional work is I used I built a phylogeny, a maximum likelihood phylogeny, by extracting RBCL and MATK sequences from GenBank, and then they were aligned in CView and trimmed in Mesquite and concatenated, and then I used IQ tree to build the existing phylogeny you see here. For the functional dendrogram, being that some of the functional traits were not available using the databases that I had, I constrained the functional relationships to the top 25 most common species across the sites. And that's the left diagram here. And the traits that were associated to build this dendrogram were leaf nitrogen content, specific leaf area, plant height, and then a categorical trait, which was growth form. So looking at correlations, there seems to be a very interesting set of observations where diversity metrics were positively correlated with each other. So more species sites tend to be more phylogenetically and functionally diverse. However, they were not significantly correlated with patch characteristics. However, patch characteristics themselves were significantly correlated with one another, which indicates that larger sites typically have larger perimeters. And then this AP here is a ratio between area and perimeter, which is kind of an indice for talking about how regularly shaped that patch is. So larger sites tend to be more regularly shaped. And then looking at average leaf nitrogen content, specific leaf area and pH, we're seeing that at least with SLA and pH here, that larger sites tend to have taller trees with greater SLA, which means that sites that have more of these bigger woody species tend to be less diverse compared to more open, larger patches. And then looking at the regression analyses based on total species richness as well as metapopulation species per site, we can see that there is a positive nonlinear relationship where taxonomic diversity is driving phylogenetic and functional diversity, at least in unstandardized phylogenetic and functional diversity. And what I mean by that is that these measures are somewhat dependent on the number of species in the calculation of these different formulas. However, when we use standardized versions of phylogenetic and functional diversity that kind of don't account as much for species richness, we can see that those relationships are not as prevalent or significant, where the only case where there was species richness was somewhat driving one of these indices was with mean phylogenetic distances. So MPD is the distances based on the phylogeny, and MTD is the distances based on the dendromic, the functional dendrogram. So to discuss this, despite the harsh conditions that are in these sandstone outcrop communities, they still are quite diverse. The fact that taxonomic diversity is positively correlated with phylogenetic and functional diversity seems that these communities are not necessarily saturated. And based on the species, the correlations, it seems that these sites are not assuming the typical species area relationships across the sites. Metapopulations tend to be short-lived exotics, composites, or some form of grass or graminoids. Species-rich communities also tend to have more metapopulation species, significant metapopulation species, and that tends to indicate that these metapopulations are able to get to a site and colonize, and they're not competitively excluded. So meta-communities are driven by environmental gradients that facilitate species turnover, and that unstandardized phylogenetic and functional diversity tends to be driven by total and metapopulation species per site. And these positive relationships between all these diversities indicates phylogenetic inertia, where more phylogenetically diverse sites also tend to be more functionally diverse. So moving on to chapter 3, looking at a larger, broader grouping of natural zodiac forest openings. These sites included chert, sandstone, shale, loess and limestone sites. Natural xeric forest openings I predicted were going to remain quite stable and resilient over the past 30 years, and that environmental diversity and disturbance-based variables would explain differences in composition based on substrate type. And so next, I wanted to see if certain species are significant indicators for specific substrates or surveys. And then my last thing I want to look at is that there is a distinct phylogenetic and functional signal observed across NXFO communities based on substrates and surveys as well. So looking at the surveys, I brought in the spatial aspect of this to include 19 different natural xeric forest openings, as you can see here. And so in this case, I only recorded a vascular taxa, so it was uniform with Heichen's survey that she did in these sites in 1988. And following that, there were several taxonomic indices that were calculated, as you can see this list here. And then after those surveys, it was seen that across both surveys, 242 taxa were observed for all the 19 sites. And overall, species richness increased for most sites between those two surveys. The only cases where it didn't change, it didn't decrease, the species richness remained the same. So most sites got more species. And then similar to chapter 2, I built a phylogeny based off of maximum likelihood and calculated 3 different indices to understand relationships based on phylogenetic clustering and overdispersion, which is this P, NRI, and NTI, where positive values of this indicate significant phylogenetic clustering, and lower numbers of this indicate phylogenetic overdispersion. Similarly, with the functional dendrogram, I limited this to the top 25 taxa that were observed in each survey, and being that 4 were exchanged in that time, this dendrogram represents the top 29 of those taxa. And then similarly with the phylogeny, I also calculated phylogenetic nears, NRI, as well as phylogenetic NTI, which are the same general relationships where if it's positive, that means that there's functional clustering. And if it's negative, it's functionally overdispersed. So after doing all that work to get those different aspects of diversity, I generated taxonomic, phylogenetic, and functional non-metric multidimensional scaling ordinations. And for each of these that I'll be presenting today are based on those top 29 dominant species. So it is based on the same set of taxa. And then I used repeated permutational analysis of variance and permdisp to understand if there are differences in composition based on site location within the ordination, as well as the overall dispersion, whether or not there was more variation within a certain substrate type or across surveys. And then since those are global tests, I used a pairwise Adonis test to determine which of these substrates were significantly different from one another. And then to understand which variables are explaining community composition differences the best, I used a vector analysis. And then to double check to see if these different TNMDS, PMDS, and FNMDS ordinations were significantly different or not from one another, and used a Procrustes and protest analysis. And then finally, I used an indicator species analysis to determine if there are differences between substrates and surveys. And so this list over here in the bottom right-hand corner, these were all the things I tested using vector analyses. So that's the list that I used for what we're going to see next. So based on all of these ordination, there were no significant interactions between substrate or survey. So now looking at the individual factors, there were significant differences in these different groupings of natural xeric forest openings based on their substrate, but they were not significantly different based on surveys. So they have remained compositionally stable and resilient across time. And to explain this ordination here, the red is sandstone, yellow is shale, blue is blue is limestone, and then the green is loess. And the lighter shade is from 1988, and the darker shade is from 2019 when I did the surveys. So looking at the vector analyses based off our squared values, The variables based on environmental conditions that were the most determinant were canopy cover, pH, photosynthetically active radiation, and soil depth. And so what this is saying, what's largely driving differences between these communities is whether they have more of an open canopy and if more light can get to basically the substrate level. And also with soil depth, lowest hill prairies typically have very dense soils due to lowest deposition. So that explains that compositional dissimilarity. And then for diversity, phylogenetic measures tended to explain a lot of the differences between individual sites where basically the sandstone and shale sites typically had greater levels of phylogenetic diversity, whereas the loess and the limestone sites had lower levels of phylogenetic diversity. And then finally, only one measure of disturbance, which is exotic species richness, was significant, and that tended to favor the sandstone sites. So moving on to indicator species, there were 87 species across the 242 taxa observed that were specific indicators for specific substrates. And then 13 indicator species were observed based on the two different surveys. And so I'm just going through here. This is the taxonomic, the phylogenetic, and the functional ordinations that were calculated. And these vectors were high in their R-squared value across all these ordinations. However, all the ordinations were significantly different based on Procrustes and protest analyses. So looking at phylogenetic and functional signal, there was no phylogenetic signal observed across all of the sites based on substrate survey, based on all the different functional traits, as well as based on the relative abundance of individuals across site. However, there were 3 specific cases of significant functional trait signal based on dominant communities as top 29 taxa. Shale communities across survey had functional signal. Sandstones communities also had this. And then finally, sandstone communities appear to have a higher functional signal during the later surveys. And what this dendrogram shows here are these top 29 taxa, and the color of the nodes indicates if they are a significant indicator for specific substrates, and it's color coded based on the NMDS ordination groupings. So despite simultaneous anthropogenic threats to community assembly across these sites, NXFO communities tend to remain stable and resilient, at least as of far. Both communities and indicator species remain distinct, but they do appear to be dependent based on their substrate or their edaphic conditions. And the variables that best explain the differences between these communities are phylogenetic measures, where sandstone and shale sites typically have greater levels of phylogenetic diversity, and limestone and loess communities tend to be lower phylogenetically diverse. Community variables are showing that limestone and loess sites tend to be more open, with more radiation getting to the substrate level, whereas the sandstone and the shale sites are more closed and typically are more shaded, more or less. And then while no phylogenetic signal was detected across sites, certain NXFO communities are starting to exhibit, at least for now, some kind of functional signal over the past 31 years. And I think a lot of this functional signal based on the analysis appears to be that there's a lot more oaks and different kinds of hardwood trees that tend to infiltrate certain communities. So looking at my last chapter here, this is looking at temporal changes in the spatial scale and the autocorrelation structure based on different patch characteristics. And I had only 2 hypotheses for this chapter. One is that diversity indices will exhibit significant spatial autocorrelation based on these natural xeric forest opening communities. And the second hypothesis, there's going to be significant taxonomic, phylogenetic, and functional turnover between these sites that are dependent on edaphic, climatic, and elevation variables. So the methods for this is that basically the survey data that was used for chapter 3 was incorporated into this analysis as well. So those are repeat methods. And then site centroids for all these individual natural xeric forest openings were included to have a spatial data frame. And then to look at global autocorrelation structure, I used Moran's eye for all the different diversity, climate, and soil variables. And those variables came from PRISM, Chelsea, as well as soil grids for all those different types of variables. And then for the individual variables that exhibited global autocorrelation structure, I constructed spatial autocorrelatiograms to understand how these different variables spatial structure change based on distance from individual sites. And then finally, I used generalized dissimilarity models to construct taxonomic, phylogenetic, and functional turnover models for both 1988 as well as 2019. And this dissimilarity matrix was based on Bray-Curtis, similar to the NMDS ordinations we saw earlier. So based on global Moran's eye, the only soil variable exhibited any autocorrelation structure was soil depth, as you can see in the top diagram here, where based on the autocorrelatiograms, at closer sites, they tended to exhibit similar values in their soil depth. However, when you get to broader scales, that pattern tends to become more overdispersed, as you can see here. This is spatial clustering, this is spatial overdispersion, and this is the area of insignificance. So several diversity variables and climate variables did exhibit significant autocorrelation, with most of them also, like soil depth, being clustered at local scales and becoming either insignificantly structured or overdispersed at larger scales. These variables here are the ones that are significant. So there are several measures of taxonomic, phylogenetic, and functional diversity that exhibit spatial structure, and then temperature and precipitation are also significantly structured. One interesting thing here is that so far as precipitation in 1988, it seems that there's spatial overdispersion for precipitation in 1988. However, that spatial structure shows now that that's actually clustered. So there's some change in the the precipitation across those two surveys that are manipulating spatial structure. And then the GDMs that I ran the 6 different models, as you can see here. And this table here shows the rank of importance of individual variables that were included in the model after backward elimination. And consistently, as expected, geographic distance consistently was the most influential predictor of biological turnover. So sites that are further away tend to be more compositionally different. However, there were differences between the different models based on aspect of turnover. So, however, 2 variables remained important across all the different models, which were soil bulk density and temperature annual range. There were a couple other interesting relationships as well. For taxonomic turnover, it seems that pH index seemed to be an important predictor. For both taxonomic and phylogenetic turnover, precipitation of the driest quarter and organic carbon stock were important. And then for functional turnover, cation exchange capacity and mean temp of the warmest quarter were important for predicting turnover. Overall, across all these models, based on deviance explained, it seems that functional models tend to be the best at prediction of compositional turnover overall. And during the later surveys, the models overall performed better compared to the models from 1988. So based on this, soil depth was the only soil variable which exhibited global spatial autocorrelation along with several diversity metrics. And I do think that the reason soil depth had significant spatial structure has a lot to do with glaciation events with lowest deposition. Over time, those sites gained basically deeper soils due to that deposition. And then most of the spatially structured variables across all these sites showed clustered or similar values at local scales and even And then at larger, more regional scales, there was either no structure observed or they tended to be more over-dispersed. Finally, geographic distance, climate, and soil variables best explain functional turnover compared to taxonomic and phylogenetic turnover. And then predictor variables that most ubiquitously explained turnover across all the sites across all the southern Illinois extent were soil bulk density as well as temperature annual range. So general conclusions here, it seems that local sites, most of the species that are getting exchanged between different patches are often short-lived exotics, composites, or grasses compared to other types of species. And these meta-communities are characterized largely by regular species replacement driven by environmental conditions where species are replaced almost as groups as opposed to individualistic species responses, indicating that a lot of the species in these sites react physiologically in a similar way to environmental conditions. At regional scales, looking at a broader group, natural xeric forest open communities at current are still stable and resilient despite anthropogenic threats such as climate change and exotic invasion. Across several different aspects of biological turnover, the things that best describe the biological turnover across these sites are geographic distance followed by bulk soil density and temperature annual range. And so this graph that I'm showing you here is reminiscent of the graphs I originally shared with you guys to kind of understand how these different diversity relationships basically explain what community assembly drivers are the most influential for community assembly, where with chapter 2, you can see that the relationship that we would expect is significant. However, there does seem to be some sites that sit on either side of this line, where in this example here, B shows that there's greater competition between clades, whereas E, intercladal competition or environmental filtering tends to be driving that community assembly. And then based on these, you can see that these sites fall above this line, indicating some kind of niche participating or limiting similarity, whereas the sites below the line are indicating some kind of a filtering mechanism that's largely driving those assemblages. And then based on the relationships between based on phylogenetic inertia, that relationship is significant, but there are again, individual sites that show functional surplus and functional deficit. And then looking at the regional scale where we look across several different sites, we can see that the sandstone and the shale sites here tend to sit on the side of competition within clades, where at the other end, there tends to be phylogenetic underdispersion indicating biotic filtering. And those relationships are seen more throughout here, where specific colored dots indicating different substrate types tend to fall on one side of that line or the other. So these different communities are being driven by different kinds of community assembly mechanisms. And then finally, so across scales, the typical species area relationship that's kind of taught to us in ecology, this relationship isn't observed across these naturally patchy ecosystems at any scale or for any aspect of diversity. And so looking at all these hypotheses together, the first 3 of chapter 2 are accepted, being that 15% of the taxa exhibited significant metapopulation dynamics. There were coherent regular species replacements based on the second hypothesis, and that diversity characteristics and patch characteristics were positively correlated with each other or within themselves, but they were not significantly correlated with each other. And then finally, nonlinear regressions show that while unstandardized measures of phylogenetic and functional diversity are driven by species richness and metapopulation species, standardized measures don't follow the same pattern. And so for the third chapter 3 here, which looks at that regional scale, natural xeric forest open communities remain resilient despite anthropogenic threats. And the variables that best explain their dissimilarity are based on light soil conditions, their phylogenetic diversity, as well as their dominant species richness. And then the one disturbance variable that tends to be explaining their compositional difference is the number of exotic species that occur within an individual site. Several indicator predictors were observed for individual substrates and surveys. And then finally, while there was no phylogenetic signal detected across all of these sites, there was a functional signal associated with sandstone and shale Mesozoic forest openings. And then for chapter 4, looking at spatial analysis, it was observed that there were specific taxonomic, phylogenetic, and functional indices that exhibited spatial autocorrelation structure. And so that was correlated with that, was structured as well. And then finally, this is a slightly accepted hypothesis here. Taxonomic, phylogenetic, and functional turnover were dependent on edaphic and climatic conditions. However, elevation was never a significant predictor across all these turnover models. And with that, I would like to thank my doctoral community, Dr. Gibson, Newbig, Sipes, Brown, and Holzmuller. I'd like to thank the Gibson Lab past and present. You guys have helped me out a lot with my research. And then these individuals here either helped out with field surveys, plant identification, drone imagery, advice, emotional support, and then a couple of societies that helped. Illinois Native Plant Society was funded the second 2 chapters of my research. So thank you for that. And then finally, SIU Plant Biology for taking me on as a student for 6 years. And then thank you to all of you for showing up and being supportive. Starting a PhD during COVID, it's a different beast. And I'm just glad you guys were there. So, and I will accept any questions.